Let me introduce Dr. Glen Pyle, who is the second speaker today on his research. And Glen started his career at the University of Guelph when he was an undergraduate student in the Human Kinetics program. How many people are undergraduates in this class, in this room? Okay, so Glen has a very important message on experiential learning. So after he did that HH&S degree, he worked for the Blue Jays, and in the course of which almost became a professional catcher for the Blue Jays. But just before he signed on, he came back to do grad school. So I think there are multiple messages there about the path for research. So Glen did his doctoral work at the University of Tennessee Health Sciences Center and held an American Heart Association postdoctoral fellowship at the University of Illinois. Since joining the University of Guelph in 2003, Glen's lab has enjoyed uninterrupted funding from CIHR, the Heart and Stroke Foundation, NSERC, and the PET Trust. Glen studies the causative mechanisms of heart failure as well as novel treatments, and I believe is currently studying how hormones regulate heart failure. Glen's research has been recognized with a number of awards from organizations including CIHR, the Heart and Stroke Foundation, and the Premier of Ontario. So I will pass it on to Glen. Thank you.
All right, so we'll jump right into this. I'm going to focus today on our research looking at the relationship between sex and cardiovascular function, cardiovascular disease. But I thought I'd start off by giving a little bit of an overview to show you where this fits in the context of our research. So what's going on in my lab is we have a number of different projects which come together to create a heart failure program. That's the focus of our research. And we're looking at everything from focusing on projects that focus on signal molecules, in the case of CAP-Z, focusing on a protein and its therapeutic potential, but also looking at clinical applications, dogs with naturally occurring heart failure, human heart failure patients, and looking at the molecular changes that occur there. Although the work is run out of our lab, we rely on an extensive network of collaborations both here and abroad. So everywhere you see University of Guelph up here, that denotes a collaboration with someone else outside of our lab, but here on campus. And then you see other collaborators across the country and really throughout the world that really make this work possible. Even though each project is distinct and in many cases has its own distinct funding stream, they do interact and we occasionally see crossovers. So for example, later on this evening, I'm giving another talk across town, so I'm trying to keep these talks focused, so if I veer off, that's why, where we're looking at the effects of sex and aging on cardiovascular changes and how the heart changes, and targeting the protein CAP-Z to potentially slow the aging process. So we also see these interactions between our projects. We follow the disease or the treatments from the level of the molecule, the protein, all the way up to the whole animal. So we look at protein changes and we look at how this affects the animal, whether that's a mouse or whether that's a dog. We're interested in the range of explorations here. So this was a slide intended for the dean, who I believe is not here, because I wanted to point out that when we integrate these projects, when they cross over, and we collaborate with other labs throughout the world, we're able to follow things from the molecule to the whole animal, veterinary medicine, human medicine, basic research in there. It really falls nicely under the One Health umbrella, which is really a strength not only of the college, but of the university. So we're focusing in on heart function, how it changes in disease, and today I'm going to talk about the relationship between sex and cardiovascular disease. So first of all, why cardiovascular disease? Obviously, it's a leading cause of death worldwide, so it's a significant problem. But what you may not know is that cardiovascular disease is no longer considered to be a disease of the Western world. So this was the tag, it's a disease of the Western world, whether that's a good term or not, that's how it was tagged. This is no longer the case. It's truly an international problem, and we're starting to see it increasing and rise to the levels that we are used to in Canada and other parts of the world. So for example, in China, So last year I went and gave a talk over there because they're investing more effort into understanding cardiovascular disease because of the significant increase in these diseases within China. Whereas it used to be in very low levels, it's now much higher. So we're seeing it spread really throughout China. To illustrate that point, if you look at cardiovascular mortality in China, really over the last 10 years, you see it's gone up dramatically. It's increasing at a rate of about 25% per year and is projected to continue to do that for a number of years. So why is this? Why is cardiovascular disease, why does it account for almost 50% of all deaths in the country? First of all, there are a tremendous number of smokers there, which you'll see if you go over. It's widespread. But in China, as in many other parts of Asia, we're seeing increased rates of diabetes. Over half of Chinese are pre-diabetics. And because diabetes is a comorbidity or something that leads to cardiovascular disease, this is driving a significant portion of the increase in mortality rates. So that's why cardiovascular disease. But today what I want to do is talk about the relationship between sex and cardiovascular disease. And so for those of you who may have seen some of this on social media, I may have mistakenly misled you to think that what I'm going to talk about was sex, the procreative or the recreational activity, and that's just to get people hooked in. But for those of you who are interested in how that sex influences your heart, well, quite frankly, it will eventually kill you. And that's shown here in this article in the Toronto Star, and of course we can't always believe the popular press, but there is actually evidence to back this up. If you look at individuals who are in the process of having sex or who have recently had it, your risk for having a heart attack will go up dramatically. So take from this, they did a whole study on this, but you can just sort of think back to what your mom told you. Don't do it, it will kill you. That's sort of the bottom line. But that's not the sex that I'm talking about here. We're talking about the differences between males and females. So social scientists often refer to this as gender, so that's perfectly fine. But in the biological sciences, we refer to these as sex differences. And we're going to deal with these in a binary fashion. I understand there is a continuum there, but we're going to deal with males and females and how they respond differently to cardiovascular disease. So why sex? Well, again, cardiovascular disease has been considered for a long time to be a man's disease. When you think about who it is that gets cardiovascular disease, most people will think about middle-aged or older men. That's the prime target. And while that's true, they have high rates. Increasingly, we're seeing that the face of cardiovascular disease is not the middle-aged or the older man, but it's increasingly female and older females. They're accounting for a greater percentage and have worse outcomes than the males. So to illustrate that point, look at this curve, and I could have taken data from a number of different places. If you look at the advances that we've seen in cardiovascular research over the years, you can see that the mortality rates for men in blue there have gone down significantly. This is a result of better research, better medicine, better management. And yet the female curve really doesn't change. It definitely doesn't go down to the same level that it does in men. So we have a huge discrepancy in our understanding of how cardiovascular disease develops both in males and females. Certainly there are non-biological effects here. There are differences in access to care and the intensity of care and things like that. But there is a biological basis for this, which quite frankly we don't understand. Now, one area where we do understand or have a significant amount of evidence that focuses on women is a hypothesis or a theory called the estrogen protection hypothesis. And this is illustrated here. We have known for quite a long time that at younger ages, females tend to have lower rates of cardiovascular disease, better outcomes than age-matched males. Now, as both cohorts age, the risk goes up, but it goes up more significantly in females, so that protective phenotype that they see earlier disappears and they may actually be worse off at older ages. And just looking at the timeline of this, the ages when this happened, people noticed that the switch seemed to correlate with the onset of menopause. So that in the earlier cohort, when they had protection, there was higher levels of estrogens, and as those estrogens decline, the risk goes up significantly. And the hypothesis was advanced that it's those estrogens early in life that protect women. if this was true, then if we introduce it later in life, they should regain that protection. Initially, this hypothesis proved to be true, or at least the early evidence was very positive, and we saw all sorts of studies coming out showing that women given exogenous estrogens, given hormone replacement therapy, had lower rates of cardiovascular disease, better outcomes, and so forth. So that at one point, hormone replacement therapy was the most widely prescribed regimen in the world. It was overwhelmingly positive. But as more research was done, we started to uncover problems, and the clinical evidence actually shifted away from this. And what we end up with is a significant body of evidence on both sides, supporting protection and supporting damage, that really just don't go together. So I'm going to leave it at this point, because if you've ever been to a conference where people talk about hormone replacement therapy, women's health and cardiovascular disease and so forth, if we get into the debate as to why this happened, these discussions become very heated and sometimes very personal, so I don't want to open that up here. So we'll just recognize that there is this discrepancy and we don't understand that, because that's a point that both sides agree on. We can't reconcile these differences and we can't understand why these things happen until we do more research into understanding, first of all, how do estrogens work and how do they work in the cardiovascular system. Seems like very simple points, and yet we don't really know how they work. So think about that. We're giving treatments to older women to mitigate cardiovascular disease, and we don't really know how it is that they work. So this has caused some drive to go back to the bench, to go back and look at basic research and find out how it is that estrogens work. As a basic scientist, that's something that, of course, I'm all in favor of. And while there certainly is moral support for this, unfortunately, there hasn't been a lot of financial funding or financial support that goes along with it. So this is my subversive political comments here, that if you want to see more research like this, you should support the report and tell your MP that you support more basic research, or more funding for basic research, and more funding for things like women's health. So that's my very subtle political thing. So we have gone back to the bench. We've always been interested in how hormones regulate the heart, but we're going back and asking what seem to be very simple questions, and they are, but you'll see as we go through them, we really don't know what the answers to these are, and they form the basis for something that will eventually be more complex and translate back into the clinics. So we want to know, how is it that the receptors that estrogens work through, how do they affect the heart? Again, you would think that before you gave this sort of treatment to a group of people, you would understand how it works, and we don't. How does menopause impact the estrogenic response of the heart? So it's possible that it's protective early in life because the heart has a certain conformation, and that as women go through menopause, the heart changes, and the ability to respond to that differs after the menopausal period. Again, a fairly fundamental question. So the way we address these questions, we have a couple of different approaches. I'll go through them fairly quickly, but just so that people understand our approach. So we have a system in the lab called the Langendorff apparatus that allows us to perfuse the hearts of mice and maintain them functioning. So if you haven't seen a mouse heart, you look at your baby fingernail, that's about the size of the mouse heart. So we take it and we attach it to a needle here, so that goes through the aorta, so the heart can be perfused and kept alive. It's mounted on the apparatus and tied in place, so that kind of gives you an idea of the size there. And then it's instrumented, so a pressure transducer goes into the ventricle so that we can measure how it functions. So this is what the system will look like. Down the bottom, you have the heart. It's in the temperature-controlled environment, and it's perfused. Now, I have an image here showing it beating. The heart actually beats at about 450 to 500 beats per minute. For whatever reason, this is very slow, this movie. So you can see the heart beats there, and as it's beating, it's compressing on the transducer. We're able to measure the functioning of the heart. Now, what we can do when it's there is we can treat it with drugs, we can stress it, we can do all sorts of things. And what we wanted to do here was treat it with drugs that activate the different estrogen receptors to see what happens. Again, this hasn't been done before. No one knows what these receptors do. In the heart, there are 3 different receptors that the estrogens work through: alpha, beta in green, and then in orange, what's called GPER or GPR30. So all of these are present in the heart. They provide targets for estrogens to interact with. And what we want to know is, what does each receptor do when it's activated? What you can see is, so we have measurement of contractility on the y-axis, and we have time on the x-axis. Take away 2 points from this. One is that if you look at the curves down the bottom, you can see that they all have a depressive effect, but they are all slightly different, which suggests that even though they're all estrogen receptors, they have different mechanisms, they have slightly different effects, and certainly the timeline is different between them. 2, if you look at the timeline, this is done in minutes. For people who have taken their undergrad biology and you learn about sex steroids or these hormones and how they work, typically they work through genomic mechanisms, which means in order to produce an effect, they have to change gene expression. So the gene has to be turned on or off, the protein levels have to change, it has to be inserted into wherever it is, and then you get the functional impact. This normally takes hours, potentially days, and yet here we're seeing effects in under 5 minutes. In fact, the original study done by one of my students, Justina Culpa, looking at ER alpha activation, we saw effects within seconds. So we put the agonist on the heart, saw a very rapid effect. So these effects are not just genomic, but they're non-genomic, and they work very quickly, and they have significant impact on heart function. Those were hearts from female mice. These are the males, because males also have estrogen receptors, and we see a different response. So it's a little more clear when we lay it over top here. So if you look, for example, at the green lines here, the dotted line is the male response, the solid line is the female response. And what you get from this is that in general, the response for the male hearts lags behind that of the females. And most people would say, well, that of course makes sense. Males don't have as much estrogen, so they of course wouldn't respond the same way. Except keep in mind, we've removed the estrogens here. There are not estrogens impacting the heart. These are drugs that specifically activate the receptors. And the receptor levels in the hearts are actually the same. We do not see, and other groups have not seen, differences in estrogen receptors between males and females, which means the target is the same between males and females, and for some reason they respond differently. So estrogen receptors depress the heart, they have a sex-dependent difference, and we don't know how either one of these work. And tomorrow I will submit an NSERC application that hopefully will address that, some of these problems. This just lays out the baseline for how the heart function and how estrogens may regulate it, but we're interested in how menopause may change both heart function and the ability to respond to estrogens. And so we use a unique mouse model to undertake this research. Historically, studies that looked at or attempted to look at how menopause affected heart function or a variety of other functions use something called ovariectomy, where the ovaries are surgically removed from the animal. This creates an estrogen deficient environment, as you would see after menopause, but it's a sudden onset, so it doesn't have the same timeline, and it lacks the menopausal phase, so we don't have that transitional phase. Now, about 10% of women enter menopause through some sort of surgical procedure. The ovaries are removed for whatever reason. So this does represent some segment of the population, but the vast majority of women go through a transitional phase that's not captured here. About 10 or 15 years ago, Patricia Hoyer's group at the University of Arizona developed a new model where they injected, you can do it in mice, rabbits, monkeys, they've done it in all sorts of different animals, with a compound called VCD. And the VCD very specifically targets the ovaries and knocks out their estrogen producing ability. It leaves intact the other parts, so it's still able to produce, for example, androgens, which is lost in the overectomized model, but the estrogen levels go down, and they go down gradually. In fact, the changes actually capture very nicely what we see in humans. So if you look at the top slide, these are some of the hormonal changes that they see in mice, or at least a representation of what goes on. And down the bottom, you see what happens in women, and you see some similarities between these two. But one of the points that we found particularly interesting is this menopausal phase, where they go from being pre to postmenopausal, and it's a gradual transition. So this model allows us to study that phase, which has never been studied before. So we implemented this model in our lab. We talked to Dr. Hoyer and got some advice on how to do this. And we injected some mice, and there's a few points along the timeline I want to draw your attention to. Around about day 40 of this experiment, the cycling of the mice becomes prolonged. So their estrous cycle is normally about 4 days. We start to now see cycles of 5 or 6 days, but they're reasonably consistent. Again, something similar happens in women. Around day 60, timeline is different though. Around day 60, we start to see pauses, which means they cycle and then they stop cycling for 1 day, 2 days, 3 or 4 days, whatever it is, but then they go back into the cycling. So it becomes discyclical. Between 90 and 100, they stop cycling. So that by 120 days, they're no longer cycling, and we've reached the end of the menopausal period. So we wanted to know what happens to the heart, both on a whole organ level and on a molecular level during this phase. Again, no one had ever bothered to look at this before. So we did echocardiography, which is essentially ultrasound of the heart. This is done in collaboration with Dr. Martino's lab. Take an ultrasound probe, you hit the heart, which is in the animal and functioning with sound waves. We're able to determine structure and function. So we're able to see what the heart looks like in the animal and how it functions. Bottom line, we didn't see anything. Now that might at first be disappointing. I mean, we always want to see some sort of crazy result that we can then publish. But the truth of the matter is, this is actually what we were hoping to see. Because if you look at women who enter menopause, they have heart function of whatever parameter you want to measure. And then when they come out the other end, those parameters are relatively similar. We don't suddenly see postmenopausal women dropping off into heart failure. So the fact that we don't see any sort of disease process here tells us that while the heart may be altered because of the molecular or because of the hormonal environment, it's not diseased, it's not damaged really in any significant way. So this recapitulates what we see in women. But just because the heart's functioning normally doesn't mean the components that contribute to its function are functioning normally. We see this in heart failure, that in the early stages of the disease, heart appears reasonably normal, but underneath is kind of a house of cards, and it's going to collapse, especially if you put some sort of stressor on top of that. So could this explain why postmenopausal women have, in part, poor outcomes? Because while the heart appears to be normal, underneath it's not functioning as it should. So the target we look at inside the cell is a group of proteins called the myofilaments. So to introduce you to what they are, here you have a heart made up of a whole bunch of different tissues, primarily cardiac muscle or myocardium. The myocardium is made up of a collection of cells called cardiac myocytes. And within those are a whole bunch of different proteins, including the myofilaments, which are shown in the middle here. This is basically the engine of the cell. This is where the contraction occurs and allows muscle to shorten and then relax. So the way this happens is an action potential is produced by the heart. It generates a signal that ultimately causes the release of calcium from inside the cell. And that calcium goes, and you can see, binds to the myofilaments and activates them. So they will then shorten, and as they shorten, the whole cell shortens. So that's the basis of contraction. It will continue to contract until the calcium is removed. Then the myofilaments are allowed to go back to the resting state and you get relaxation. So this cycle goes over and over again to generate heart contractions. So we're interested in that component of the cell, and we use what's called a Carter assay to measure how these myofilaments are functioning. What we do is we take myocardium and we basically get rid of everything else in the cell except for the myofilaments. We can then introduce calcium into the myofilaments and turn them on or activate them to whatever level we want, and they're activated across a range of levels. And as they contract, they consume ATP. That ATP is broken down and produces phosphate, and that's what we measure. So we measure the production of phosphate as a surrogate for how the myofilaments are contracting, how they're active. What you can see is if you focus first on the blue line, which is, I'll say, the normal or the control, as you add calcium, activity goes up, and then as you remove calcium, the activity will go down. So this is the contraction relaxation phases. And as the mice go through this menopausal phase, you can see day 40, 60, 120, myofilament function shifts. So to give you an idea of the scope of that shift, you see we go from blue to orange. It may not look like much of a shift, but that's a significant shift in the function. So underlying the normal heart function is a disrupted or altered myofilament complex, which means while the heart appears to be normal, it's really not. We then asked can the heart still respond to estrogen agonists as it did before? So on the left here, we have the slide that I already showed you, showing that estrogen receptor activation depresses heart function. If you look on the right, it's clear that there's an alteration here, but it's not the same. It's not simply a knockout of the response. Yes, the ER alpha response is diminished somewhat, but you can see ER beta, which had essentially the same response after 15 minutes, is now completely gone. So again, it's not simply a desensitization, it's a remodeling of the heart, a remodeling of its ability to respond to estrogens right at the end of menopause. We haven't gone postmenopausal, we haven't let these mice age, it's simply at the end of menopause. And then if we look at how the myofilaments respond to the estrogen receptor agonists, on the left you have the intact animals, on the right the mice at the end of menopause. Without going through all the details, you can see there's a difference. So the inside of the cell is also responding differently, which means the heart is not regulated in the same way that it was at the beginning of menopause. So what does this all represent when we put it together? Well, first of all, this is the first investigation of the menopausal period showing how the heart changes over time. It's not simply an estrogen deficient period where we've knocked out estrogens and said, what happens when we remove them? We're watching this gradual process and seeing how the heart is changing in real time. Heart function appears to be normal, but this is the result of altered molecular function. And this is important because we see that when the heart changes underneath, when it adapts to things, like I said before, it doesn't necessarily tolerate stressors very well. So there is certainly evidence to show that when the myofilaments, for example, are altered in a way, your risk of arrhythmia can go up, your risk of sudden cardiac death can go up, your risk of heart failure can go up, all because the myofilaments have changed. Even though the heart appears normal, it's because the inside of the cell has changed. It can't tolerate these stressors anymore. So we're wondering if this might explain some of the higher rates of cardiovascular mortality or worse outcomes. Estrogen receptor regulation is altered during menopause. So again, the first investigation showing this gradual change. And because we see a difference in how they respond to estrogens at the beginning of the phase and at the end, this might explain why it is that early in life women benefit from the high levels of estrogen, but afterwards the story is a little more complex. Simply reintroducing estrogens into an environment that may not respond the same way is probably not the best idea. That we need to better understand how it is that they respond afterwards. So what we conclude is that menopause fundamentally changes the heart, and this really necessitates that we understand how it is that this period changes the heart function, changes regulation, and how the period afterwards also impacts that. So where do we go from here? Well, we want to follow this out further and find out in the postmenopausal phase, how does the heart change? I can tell you we already have some preliminary data based on some other studies, and it's not a linear change. It's not simply that, for example, myofilament function declines over time. It's more complex than that. It drops, it comes back up, and it changes in all sorts of different ways. So it's not a linear change. Where's the therapeutic window? Some studies show benefits, some do not. This suggests that there is a target that you can hit with estrogens that may be protective, and part of that may depend on the timing. And because we don't know how the heart changes over time, that makes it very difficult to identify the window. So we're hoping to see how it changes and identify windows of therapeutic intervention. If we apply the agonists acutely, can we protect the hearts? Right? So this whole idea that estrogen receptors work, or estrogens work through genomic pathways, we don't doubt that. We're not saying that's not true, but there is increasing evidence to suggest that there are acute effects. So if we apply estrogen receptor agonists, for example, in the course of cardiac surgery or immediately after a heart attack, does that have rapid effects to protect the heart? Again, we don't know. And finally, what are the mechanisms by which these receptors may produce protection? This is important to, first of all, anticipate any sort of side effects. So if you know how it works, you can see problems coming. And we can translate this further. So the application of estrogens have problems, so they have off-target effects in terms of affecting things other than the heart. So if we can target those beneficial effects specifically in the heart, we can get benefits there. And also, and this is a very ironic statement, but if we can determine how estrogen receptors protect the hearts of women, we can now apply this to men. Okay, so we can take information that we've gained from women, apply it in men without giving them estrogens, which would have, as we would all anticipate, certainly unwanted side effects. So again, I find it ironic that we're going to do a lot of work on women or female animals and then use that to benefit the lives of men, but that's the way things go. All right, so I'll acknowledge the contributions of others in this. So Dr. Langendorff certainly has participated a lot in the Langendorff and some of the echo studies. Dell, this forms, if not all of her master's thesis, certainly a lot of it. Alex started this work looking at the VCD mice and managing about 1,000 mice over in the animal facility. So that was great. And Melissa Ferguson did a little bit of this. Mostly her work was on environmental estrogens and their effects on the heart. And of course, our collaborators in the Martina lab, Christine and Faiza, who have helped with the echoes. Thanks very much.